Good afternoon. I'm Michael Roberts, Dean of the Gupta College of Science at Coastal Carolina University. I'm here today with Dr. Karen Aguirre from our biology department. Dr. Aguirre, could you tell us a little bit about your background? And specifically, what courses do you teach?
Hi, my name's Karen Aguirre, and I teach biology at Coastal Carolina University. And the course I most like to teach is immunology, when that's the study of how the body fights off and defends itself against invaders like the coronavirus.
So in your immunology class, have you and your students been talking about COVID-19?
But in immunology, what we're thinking about really is how, again, the body responds to that virus, how the things that it does in order to clear it and return the body to health. And we're also very interested in vaccines this semester, and in the virtual lab that we've been running, the students and I have been looking at some of the structural proteins of SARS-CoV and how to identify really, really good strategies and candidates for protective vaccines against the virus.
So, tell us what happens when someone inhales a COVID virus, either that's been released from a person or they pick up from a surface.
So the virus enters the nasal passages and it goes through the nasopharynx, and at that point it's going to be going now down through the trachea and into the bronchial tubes. From the bronchial tubes, it's able to gain access to the lungs and the entire respiratory tract. Um, the initial symptoms of a person who has the virus colonizing the cells in those areas are fever, rather high fevers, uh, a hacking, dry cough, sore throat, and in some cases there is also an anosmia. Anosmia is the loss of the ability to smell, and apparently it's quite common not to be able to distinguish smells when you're suffering from the coronavirus. We don't really understand what's going on exactly there yet.
So after contact, the virus enters the respiratory system. What happens once it gets into the person's lungs?
Well, the virus grabs onto and holds on for dear life to a protein called ACE2, ACE2 receptor actually, which is present on the surface of most of those epithelial cells that are in the upper respiratory tract and even the lower respiratory tract. Um, Once it's grabbed on and is holding on tight, it's able to penetrate the plasma membrane, the cell membrane, and get inside the cell. Once inside the cell, what the virus is going to do is to shut off all of the normal cellular activities and replace them with just becoming a little factory, churning out hundreds and hundreds and hundreds of new viral progeny. So there's endless viral rounds of viral replication going on. Okay. After a while, when the host cell is just full of all of these new viruses, they burst out of the cell and they move on and infect now the next cell and the next cell and the cell next to that, until eventually the entire respiratory system is involved. Okay. At this point, the immune system is at work, and it might be doing a little more harm than good. It's trying desperately to reverse the situation. Is that you get fluid coming into the lungs, followed by lots and lots of these host defense cells. And when you have fluid in the lungs, that's pneumonia. Okay? So you have a tissue that in the lungs is ordinarily very kind of delicate and lacy and lots of air sacs. And that's replaced by this sort of swampy tissue full of fluid and lots and lots and lots of densely packed host defense cells. What happens then is that the lungs become less elastic, and you begin to have a lot of difficulty with the mechanics of breathing, the sheer mechanics. This is the point where people often are put on respirators or ventilators, which are essentially the same. And eventually this can lead to acute respiratory distress syndrome. When that happens, there's difficulty getting oxygen to all of the tissues and organs of the body.
About how long does all that take?
Well, for many people, remember that there will be no symptoms at all, and others will just experience a kind of a minor flu. Um, but for people who are going to have this kind of progression to grave disease, what we find is that there's about, say, 3 to 10 days from the onset of symptoms until radiologically it's obvious that they have fluid in their lungs. So radiologic diagnosis of pneumonia. Okay, and then at that point it can be another 6 to 14 days from the onset of symptoms to the ICU, so going into the intensive care unit, being that sick. If people do go on a ventilator, with COVID-19, what we find is that they're on the ventilator for usually like in the neighborhood of 21 days, which is quite a long period of time. That's why it's so difficult to free up enough ventilators, because people need them for quite a while. Some folks will come off the ventilator eventually and return to health, and tragically, many others will die anyway.
So tell me, why is there so much variability in how sick people will get when affected by this virus?
So load is important. Obviously, the more virus that you have deposited in your system, the more likely it is that you will more quickly go down and experience some very severe disease. Other considerations are just the health and the physiology of the person. Why is there so much variability? Well, is the person very healthy and young and exercises a lot? Does the person have maybe allergy or asthma? Is the person living on a street where day in and day out there's lots of traffic related pollution? Or are they in a more rural, quiet area? Does the person smoke? Does the person perhaps have some underlying syndrome like diabetes or obesity? Okay. And there's also the issue of age. Now, it's true that young people can get this virus too, okay? But you do see more frequency of severe outcomes in the elderly. And with the elderly, again, it's not all the same. Some elderly people are very active and in relatively good shape. Others are quite sedentary, and then there's the frail elderly who really have very many underlying conditions. Okay? How is their cardiovascular health? Do they take immunosuppressive drugs? All of these things will make a difference for how well they're able to respond to the virus. In addition to that, on top of that, we also have probably some genetic characteristics and anomalies that might predispose some people to resistance or susceptibility more or less to any disease, coronavirus or anything else. Okay? And we don't know about that yet. It's too soon for us to really understand much of the underlying genetics of resistance susceptibility with this particular virus. Finally, I would mention that there's socioeconomic factors that impinge on how well a person is going to respond to the virus. How well nourished are they? Are they eating a good diet? Can they afford to go and get medical attention early, or do they wait just a little too long, hoping not to have to pay or fearing that they'll have to pay a bill?
So what is the individual's immune system doing while being attacked by the virus?
Well, the immune system is hard at work while this whole thing is going on, right? So in the very early hours after infection, the immune system, the first responders are doing their job. And the entire way the immune system works is that it needs to recognize that something is foreign, that it's an invader, and then eliminate it. So the first responder cells are cells called macrophages and granulocytes, and they are right there at the site in the lungs of the infection almost immediately. And the way they work is by a combination of phagocytosis, which is a matter of just engulfing and eating the virus and then digesting it, chopping it up into a million little pieces inside the cell and killing it. Okay? Or some of these cells release and target the virus with kind of little virus poisons, if you will, some defensive molecules that we have that they're able to deploy in order to kill the virus. But it's a race, it's a war, it's a contest. And it's as much as those first responder cells can do sometimes, just to kind of hold the virus in check and have it not explode and get entirely out of control. In the meantime, while all this is going on, what's happening is that the second wave of response is being prepared and developed and begins in the bone marrow and continues in the lymph nodes. Okay? And so there's a certain kind of stem cell that lives in the bone marrow, and it gives rise to lymphocytes. And what will happen is about 7 to 14 days in humans after the onset of infection, you have a nice developed army of very specific, dedicated B and T cells, which are going to fight that infection. The B cells by making antibody, which is very good at neutralizing virus if you can catch them when they're moving in between cells and they're accessible to antibody. And then the T cells. What the T lymphocytes do is they secrete a lot of cytokines, which are immunochemicals. And the cytokines help to stimulate the B cells and those macrophages to get angrier and angrier and angrier and work even better at their job. But the other thing that the T cells can do, which is super important, is that the T cells can recognize, they can tell when there are virus hiding inside one of the host lung cells and replicating. They can tell that from outside. And then so what the T cell does is it kind of cozies up to the infected cell, and for the good of everybody, it kills it. Okay? The Bs and the T cells acting together are very fierce and effective warriors. And eventually what happens almost all of the time with infection is that they win. Game over, the virus gets its butt kicked, and it gets kicked out of the immune system. When that happens, the giant army of B and T cells will kind of quiet down and die off itself, except that you get left behind a little subgroup of what we call memory cells. The memory B and T cells will continue to be very long-lived. Some of them live for the life of the entire organism. Some of them live for a few years, 5 years, 10 years. And what they are is they're primed effector cells that are ready on a moment's notice, if you ever encounter that virus again, to go to war against it just as hard and as strong as it did the first time, but without that 7 to 14 day lag. And the result is that you're effectively immune to further encounter with that virus. It's not that you'll never ever see it again. It's that your body, your B and T cells, those memory cells will handle it so quickly before it gets out of hand that you won't ever even know that you were infected again. And that's immunity.
So in the whole infection process, when exactly is the individual contagious?
Well, a person can spread virus even if they're not symptomatic. At any point from when they're very shortly after they're infected, they become able to spew out virus really in their saliva. So that's why social distancing is so important. Okay? And that's why it's so important for us all to follow the guidelines of the CDC and to wear some kind of a cloth mask over their face when they go out in public, when they leave their home. The reason for that is that it really stops a person who may not know they've been infected, and that could be me, and that could be you. We don't know. The mask stops us from spreading the saliva all over the place, right? So it's really more that it stops us from spreading the disease than that it protects us from inhaling, okay, through that little piece of cloth. But if you think about it, if everyone does this at once, then it's kind of a kindness that you do to the other people that you meet, that you're covering up so that you don't get them sick if in case you have the virus. And if they do that, then it's a reciprocal kindness, and everyone is protected. And that's what we're looking for, right? So the social distancing is very, very important right now. And wear your cloth mask. And of course, all the things we say about washing our hands frequently and wiping down surfaces, these are all important. It's also important to stay healthy. Try not to stress too much. Eat well, sleep well, get a little exercise. In short, we need to take care of ourselves, and we need to care for the others around us as well.
What drugs might be available to fight COVID-19?
Well, the short answer is that we really don't have a great antiviral drug right now for SARS-CoV-2. There's been a lot of buzz about hydroxychloroquine. Now, hydroxychloroquine would have to be repurposed. It is a malarial drug and a drug that's prescribed for people with lupus, the autoimmune disease, SLE. Okay. So it turns out that in a few small trials, chloroquine was helpful. Hydroxychloroquine, excuse me, it's a related drug. There are two of them really, hydroxychloroquine and chloroquine. Anyway, in some cases, it has proved to be helpful in somewhat mitigating the effects of the disease. And so it may be somewhat protective for people who are already ill. Okay. Unfortunately, the data that we have so far is still too spotty and not systematically and extensively enough gathered and reported that immunologists or doctors feel a great deal of confidence in hydroxychloroquine as a drug. Another problem with it is that it is not tolerated, and so it cannot be prescribed for anyone who has a kidney or liver problem because it can lead to cardiotoxicity for them, and you cannot give it to people who are immunosuppressed. Okay? That said, who knows, maybe it will turn out to be a good part of our arsenal. In addition, there is a drug called remdesivir. Remdesivir is an antiviral, and it was developed for to fight Ebola and does very well against Ebola. Now, it turns out that in some tests in vitro, so in the petri dish, remdesivir also does very well against all three of the coronaviruses. So that's SARS-CoV from the initial coronavirus epidemic of 2003 or so, MERS, which is Middle Eastern Respiratory Syndrome, which is also a coronavirus, and the third, which is SARS-CoV-2, the one we're dealing with right now in this pandemic. So that looks kind of promising. And we also have larger systematic trials of remdesivir that are underway. If either of these two drugs proves to be pretty good, well, we'd still need to ramp up and accelerate production and get it out to where it needs to be.
And how about vaccines? What's happening on that front?
So what you're trying to do with a vaccine is to basically try to create a situation of synthetic memory. We're going to try to trick the body into thinking it's encountered the virus and making memory cells. But really, what it's encountering is a little piece of the virus that is not dangerous to the folks who receive it, the people who are being vaccinated. So we have two pretty good candidates for a vaccine that are in very early stages of development. The first is the Moderna vaccine. And the Moderna vaccine is a little piece of messenger RNA. And the SARS-CoV-2 virus and all coronaviruses carry their genomes on a little piece, a little stretch of RNA rather than DNA. So this is just a little portion of the genome, okay? And it is the area of RNA that encodes for the spike protein, which sticks up out of the virus and gives it that spiky appearance that it has. Okay? That's a good protein to use to make an immune response to, as it turns out. So some folks are at this point, they have volunteers and they're receiving some of that messenger RNA, and we'll see how it goes. We'll see whether their body now is tricked into thinking it's under attack, bounce all the stages of infections that I told you about, going all the way through armed effector cells and winding up leaving a nice little memory component, which would then mediate immunity. There's your vaccine. Another very good candidate is really kind of interesting. What they've done is they've taken, this is coming out of University of Pittsburgh, they've taken a little bit of the spike protein and mixed it up with the sugar. And they formed those into these little spikes. And what the vaccine actually looks like is like a little patch, almost of Velcro, about the size of, maybe the end of your thumb. And there are all these little needles in an array, micro needles sticking out. But the needles, those little sharp points, are actually made out of the mixture of sugar and spike protein. And so they would put this in a good spot up against your skin, And the little spikes are going to penetrate your skin. And when they get onto the other side, what happens is that the sugar and the protein dissolves and moves throughout the skin. And in the skin, you have lots of these immune defense first responder cells that will take up the viral protein. And again, you have the whole same sequence of events occurring. We are going to eventually go through a big war, a little big fight against this actually very harmless thing that you've injected. And in the end, there'll be a nice little contingent of memory B and T cells, if the vaccine is successful, that will then be able to just be there instantly if you ever encounter the virus actually in real life and not in a vaccine situation. Both of these vaccines have done well in animal models, and now we've moved on to very early clinical trials. We need to remember that we have to gather a lot of data, make sure it's safe, effective, feasible, can be a practical thing that's actually delivered to people, cost effective, and then they will begin to actually roll it out if that's the case. But this would be probably a process of many months. The vaccine is a little ways off. Till then, all we can do is practice social distancing, keep a good thought, and we'll get through this. Thank you.
Well, thank you very much, Dr. Aguirre. I really appreciate you being with us here today.